Biochemistry 401 Lecture 32. Today we're going to continue our discussion of nitrogen-containing biomolecules. We're going to talk about the incorporation of nitrogen into biomolecules. We're going to start with nitrogen fixation and then go on to nitrogen assimilation, amino acid anabolic pathways, general synthetic strategies, and regulation. So let's get started. The ultimate source of nitrogen for the planet is actually atmospheric nitrogen gas. Our atmosphere is about 80% nitrogen. The thing is, in order for this nitrogen to be used, it must be split and reduced to make ammonium. Most of the newly fixed nitrogen, about 60%, is produced by archaea and bacteria. Other sources include lightning, ultraviolet radiation, and industry during the production of fertilizers. Now, plants take in nitrates and nitrites, and they reduce them with electrons from reduced cofactors to make ammonium for synthetic reactions. Animals like us ingest nitrogen-containing compounds and get our nitrogen supply that way. Nitrogen fixation occurs in diazotrophic organisms like some bacteria and archaea. The term diazotrophic means nitrogen fixing. These organisms can split and reduce atmospheric nitrogen, reducing it to ammonia. Now, the triple nitrogen bond is difficult to break because of the instability of highly reactive, high-energy intermediates in the pathway. The general progression from N2 to ammonia is energetically downhill. It's just getting there that's difficult because of the presence of high-energy intermediates. These reactions require special catalysts that are not found in higher organisms. Nitrogen fixation is an energetically expensive process that requires 16 ATP per mole of nitrogen split and reduced. The overall progression is N2 plus 10 protons, 8 electrons, yielding 2 molecules of ammonia, and 1 molecule of hydrogen gas. Let's see how this happens. This enzyme complex contains 2 halves, a reductase and a nitrogenase. The reductase transfers electrons from a donor protein, like ferredoxin, to nitrogenase, and the nitrogenase splits and reduces the nitrogen. Now, the passing of these electrons actually requires that the 2 halves of this complex come together, and this is accomplished through the hydrolysis of ATP. ATP hydrolysis causes a conformational change that brings these 2 halves together so that the electrons can be passed from reductase to nitrogenase. In this diagram, we see reductase on the left in white and the nitrogenase in blue and green on the right. The reductase has an iron-sulfur cluster and is called a Phe protein. The reductase contains an iron-sulfur cluster as well, but in addition contains an iron-sulfur molybdenum cluster, and so nitrogenase is called a Mo-Phe protein. The presence of molybdenum is unusual in an enzyme, and it is necessary for nitrogenase function. Now, the bacteria that carry this out can either be free-living bacteria, bacteria that live in the soil, for instance, or it can be symbiotic bacteria. Symbiotic bacteria include bacteria that infiltrate the roots of plants like legumes and form root nodules. Here, we see root nodules. Root nodules like this are seen in such plants as beans and peas and alfalfa. Now, this whole process of incorporating nitrogen is very sensitive to oxygen, and so oxygen must be kept out of the system. The sequestering of oxygen ensures that side reactions, such as the production of water, do not occur. Like hemoglobin sequesters oxygen. If you recall, like hemoglobin is a homologue of the beta subunit of hemoglobin and of myoglobin. The presence of this homologue means that the bacteria can still run the electron transport chain without endangering the fixation process. The enzymes that are involved in nitrogen flow can be a little confusing, so I have listed them here for you. We've already seen aspartate aminotransferase and alanine aminotransferase. These catalyze reversible reactions. Glutamine synthetase catalyzes the irreversible formation of glutamine, whereas glutaminase catalyzes the irreversible loss of glutamine. Glutamate dehydrogenase we saw in the last lecture, and this is an enzyme that in animals goes in one direction predominantly, to lose ammonium. We use glutamate dehydrogenase to remove ammonium from glutamate to yield alpha-ketoglutarate, and then we use the ammonium to make urea. And again, this happens, as we said, in the liver. Now in prokaryotes, especially those that are involved in nitrogen fixation, glutamate dehydrogenase goes in the reverse direction to assimilate ammonium to make glutamate. Lower organisms also have the enzyme glutamate synthetase. So let's look at this a little more closely. Here we see some of the major reactions that we're going to look at in this lecture, and some that we saw previously. Previously, we saw glutamate dehydrogenase releasing ammonium from glutamate to make alpha-ketoglutarate. We also saw aspartate aminotransferase and also alanine aminotransferase that transferred an amino group from an amino acid to alpha-ketoglutarate to make glutamate. Now we're going to look at some other reactions in which glutamine is made and broken down. The irreversible formation of glutamine occurs via a synthetic reaction that requires ATP. This is catalyzed by glutamine synthetase. Glutaminase catalyzes the irreversible loss of glutamine to make glutamate, and ammonium is released. We're going to look at this a little more closely. Glutamate dehydrogenase in prokaryotes is involved in nitrogen assimilation. In the assimilation of nitrogen, it's important that the L amino acids are synthesized, so the stereochemistry of this addition is important. The enzyme glutamate dehydrogenase catalyzes the reaction in which alpha-ketoglutarate plus ammonium yields a Schiff-base intermediate plus water. This Schiff-base intermediate is then reduced to make glutamate. This is a synthetic reduction, and so in this case we're going to use NADPH. Now when we looked at the reverse reaction in eukaryotes in preparation for the urea cycle, we used NAD+, and that was an oxidative catabolism. And so in the reaction that you see here, which is a synthetic reaction, we're actually going to use NADPH as a source of electrons. And the final product is glutamate and NADP+. The reaction catalyzed by glutamine synthetase adds another amino group to glutamate. In order to get this done, glutamate is first phosphorylated to make an acyl phosphate intermediate, and then this intermediate is amidated. We're going to release inorganic phosphate and form an amide to make glutamine. Now glutamate synthetase catalyzes a similar reaction, only instead of glutamate as the initial substrate, glutamate synthetase uses alpha-ketoglutarate. As we go into further lectures, it's important to remember this process. Phosphorylation first, and then replacement of the phosphate group with an amino group. We're going to see this when we look at nucleotide synthesis, so remember this progression. In this figure, we see glutamine synthetase. ATP and glutamate are shown in the active site of the enzyme. Now, glutamine synthetase is a highly regulated enzyme, and this is because the level of glutamine in the body is important. Glutamine is a carrier of amino groups in the bloodstream. It travels between the tissues and the liver, and it travels between the liver and the kidneys, and we're going to see this in just a minute. And we'll also see that glutamine is important in the brain. And in addition to this, glutamine is used in many synthetic reactions as a source of amino groups to produce nitrogen-containing biomolecules, such as nucleotides, for instance. And so in many ways, glutamine synthetase helps to regulate the flow of nitrogen in the body. For this reason, it's very important that this enzyme be regulated. Glutamine synthetase is a multimeric protein that's sensitive to regulation by negative feedback through its products and by alanine and glycine. Now, the binding of each of these products finely tunes the activity of the enzyme to turn it up or down a little bit, or to completely shut it down. We're going to see later that most amino acid synthetic pathways are regulated, in fact, by negative feedback from their products. This ensures that optimal concentrations are found of each amino acid in order that proteins can be synthesized, and in order that we can make amino acid derivatives that are needed also. Let's talk about the synthesis of amino acids. These syntheses use intermediates from glycolysis, the pentose phosphate pathway, and the TCA cycle. You do not need to memorize this slide. Please do not memorize this slide. It is only necessary that you understand that these three pathways are involved in providing intermediates. Now, of the 20 common amino acids, 10 are essential. Now, what this means is that we cannot synthesize them at all, or we cannot synthesize them in sufficient quantity for all life stages. For instance, we saw that we can make arginine in the urea cycle, but we do not make enough of it this way in order to support early development. So for this reason, it is an essential amino acid. Now, a way to remember this is Private Tim Hall for phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, leucine, and lysine. Now, it's important that we have all 20 amino acids that we need when we need them. If we don't get enough of the essential amino acids in our diet, this can have dire effects. The body will start breaking down proteins in order to get the amino acids that are needed. This results in a disease called kwashiorkor. Kwashiorkor occurs when there are not enough essential amino acids in the diet to support life. This graph shows the number of steps that it takes to make amino acids. The non-essential amino acids are shown in blue, and the essential amino acids are shown in red. The number of amino acids in each category are indicated on the y-axis, and the number of steps in the synthetic pathway are shown on the x-axis. We can see from this diagram that most non-essential amino acids have 4 or less steps in their synthetic pathway. The majority of essential amino acids have 5 or greater steps in their synthetic pathway. Those amino acids that are the most difficult to make are essential amino acids for us. It's much easier for us to get them in our diet than it is to synthesize them. And so whereas these pathways exist in so-called lower organisms, we've lost the ability to make these amino acids. Many amino acids are regulated through negative feedback. This is done in order that we have sufficient amounts of all amino acids, not just a lot of one type or another. And please do not memorize this slide. I'm going to walk you through it so that you understand what's going on. You might have to describe what's going on an exam if I were to give you a figure like this, but you certainly don't have to memorize everything that's here. Just understand what's going on. Let's start with hydroxyethyl TPP. This is going to be used in a split pathway. On the left-hand side, we see that pyruvate feeds in to make valine and leucine, whereas on the right-hand side, alpha-ketobutyrate feeds in to make isoleucine. Now, if a lot of isoleucine is being produced, this means that the right-hand side of the pathway is being used a lot. As isoleucine increases, it inhibits threonine deaminase to decrease the amount of alpha-ketobutyrate that's available to enter the system. This means that the left-hand portion of this pathway is going to catch up, and once the left-hand side is caught up, valine feeds into the system to turn up threonine deaminase so that more alpha-ketobutyrate is produced. In this way, both sides of this pathway are regulated so that we get relatively equal amounts of leucine, valine, and isoleucine. Amino acids are also important precursors in the synthesis of many other molecules besides proteins. These include nucleotides, phospholipids, neurotransmitters, and hormones. Please do not memorize these structures or the names, but you do need to know these 4 classes of intermediates. Another important thing that you do need to know is that glutathione is also a product of amino acids, glutamate, cysteine, and glycine. And it is the cysteine that's necessary for the antioxidant properties of glutathione. And so now we're going to talk about nitrogen flow in the body. Now, first of all, ammonium must be transported safely. It is transported from the peripheral tissues primarily as glutamine and is also transported as alanine from the muscles during the breakdown of branched-chain amino acids. Now, ammonium is released primarily as urea and also, to a lesser extent, as free ammonium in the urine. Now, in the liver, ammonium is going to be used in the urea cycle, and the excess ammonium that cannot be put into the urea cycle for some reason is going to be scavenged in order to protect the body. In the kidney, excess ammonium is going to be excreted in the urine. Now, the brain is sensitive to ammonium, but it's also sensitive to glutamate, and we'll see why in just a minute. Now, the liver has both glutamine synthetase and glutaminase. These are active in different cells. Glutaminase is active in cells that have the urea cycle, and it's used in the production of urea in order to break down glutamine to release ammonium so that it can be used in the urea cycle. Now, glutamine synthetase is used to scavenge free ammonium in cells where the urea cycle is not present. Ammonium that cannot be used in the urea cycle can be picked up by glutamine synthetase in these cells. In this way, the body is protected from increased ammonium. During periods of acidosis, bicarbonate is needed in order to increase blood pH. And because the bicarbonate is used in this way, there's less for use in the urea cycle. And so urea synthesis is slowed. But if the urea cycle is slowed down, what are we going to do with all that excess ammonium? That can be used to make glutamine. And so glutamine is made by glutamine synthetase in order to scavenge excess ammonium. This glutamine can then be transported from the liver to the kidneys, and the glutamine is broken down and ammonium is excreted by the kidneys. In the kidneys, glutaminase and glutamate dehydrogenase are important. Glutaminase is used to deamidate glutamine. Now, once glutamine is deamidated, what are we left with? We're left with glutamate. And then glutamate can be deaminated by glutamate dehydrogenase. And once we release ammonium from glutamate, what does that make? That makes alpha-ketoglutarate. The ammonium from both the deamidation of glutamine and the deamination of glutamate is released in the urine. Now, the brain is sensitive to both glutamate and ammonium. Glutamate is used as a neurotransmitter, and so it's excitatory, and it affects neuronal function. And so too much of it is not a good thing. Ammonium is also not a good thing. We know that that's toxic. And so glutamine is used as an ammonium sink and as a way to get rid of excess glutamate. Because unlike glutamate, glutamine is non-excitatory. It is not used as a neurotransmitter. And so a way to get rid of both the glutamate and the ammonium is to make glutamine. And astrocytes do this. An astrocyte is shown in the micrograph here. Astrocytes help protect the brain. They support neuronal function by using glutamine synthetase to decrease the amounts of ammonium and glutamate in the brain. And so this is a win-win situation for the brain. This marks the end of Lecture 32. Have a great day.